Welcome back to another presentation with information you'll find nowhere else. Our topic today is the bacterial species Akkermansia muciniphila. Muciniphila is a very interesting and promising bacterium and has become more popular as human research has increased in recent years, and now as it exists as a probiotic. Most commercial probiotics contain several species from the genera Lactobacillus and Bifidobacterium. These are the commercial probiotics you're accustomed to. Lactobacillus can be a good idea for infant and vaginal health, but in general is not a good idea in adults with dysbiosis, which is just about everybody. Bifidobacterium is almost universally a health-promoting genus, but you can easily increase its abundance in your gut by feeding it the fuels it loves instead of buying it as a probiotic. Muciniphila is a little bit different. When should we consider increasing its abundance, and how do we go about doing that? Here I have a summary of all the research papers I've found over the years when some type of substrate significantly increased or decreased muciniphila. If you look at this table, you'll see most of this data is the animal data. I rarely use animal data, but in this area, human data is lacking. You will also note that there are quite a few studies which showed significant increases by using various phytochemicals or plant compounds. Phytochemicals from grapes, berries, teas, and the alkaloid berberine seems to do a good job of increasing muciniphila abundance. In fact, this berberine-induced feeding of muciniphila is possibly how the medicinal use of berberines improves metabolic markers, as it's poorly absorbed in the gut. And it's well known that muciniphila is a bit of a superhero when it comes to metabolic syndrome, but more on that later. You can also see a number of trials using various prebiotics with mixed results. However, any increase here is probably the result of community changes, as muciniphila is really not a conventional prebiotic ferment, as its enzymatic machinery is much more specific to mucin, as well as phytochemical degradation. So let's talk about phytochemical metabolism. First, what exactly is a phytochemical? Well, the best example with which you're probably familiar is resveratrol, which can come from different sources, but most famously associated with grape skin. And since I advocate the consumption of aged red wine in preference to any other alcohol, if one is to consume alcohol, well, let's focus on this. In the grape, various protective plant polyphenols are produced to help it survive from insults, such as microbes and UV radiation. These phytochemicals are concentrated in the skin of the grape, which is why aged red wine has a much higher amount of phenols than, say, white wine. Although after ingestion, some is metabolized in the upper GI, but most reaches the colon where these compounds are extensively metabolized by the microbiome. As you can see in figure 3, there are many subsequent compounds which can result from the complex biotransformation of the parent polyphenol. And this is just polyphenols from grapes. Never mind the other compounds within grapes, or any of the other sources listed in the previous slide. So as you can see, what goes on inside you is infinitely more complex than you realize. This is why it's important to eat a healthy diet instead of the processed garbage most people eat. These and many other compounds are directly and indirectly beneficial to you. Resveratrol is directly beneficial as it increases sirtuin activity, which has a slew of benefits. These polyphenol metabolites are extensively fermented by our microbiome, largely by health-promoting bacteria, which increases their presence, and they in turn go on to do good things for you. Having just touched upon the phytochemical story as it relates to Akkermansia muciniphila, now we'll spend more time on its primary fuel, mucin, or more simply put, mucus. The walls of your intestines are lined with mucus, in some spots thicker and layered differently than others. This protective mucus layer is a good thing, as it keeps at a distance a mountain of molecules which can stimulate an immune response, which is critical as some 80% or more of your immune system is in and around your gut. Muciniphila has a number of features in common with opportunistic pathogens. For one, it's a gram-negative bacteria, which means it has LPS in its membrane. It's also relatively tolerant to oxygen. It can survive and even thrive in higher pHs above the Goldilocks zone for the major health-promoting bacteria, and it is able to live in our mucus as it's an effective mucin degrader. It's rare that it possesses a wide array of enzymes which are able to break down the initial mucosal barriers to degradation, and then continue degrading other bonds within mucin. But there's a catch. It can live there with other amazing health-promoting bacteria who otherwise could not thrive on their own. As it breaks down mucus, it cross-feeds these other great bacteria who in turn produce butyrate, which is the primary fuel source for the cells that line your large intestine. Butyrate does a great many wonderful things to include increasing mucin production. So you can see how this is a healthy circle of life. The cross-feeding process is very similar to how B. adolescentis and R. bromii cross-feed health promoters from their ability to degrade the bonds in resistant starch. And who are these amazing health promoters? They include names like E. rectale, E. hallei, and F. prausnitzii, and other various species from the genera Roseburia, Anarostipes, and Allostipes. These names may not be familiar to you now, but these are the true heroes of the gut. They don't exist as probiotics, but your goal should be to maximize their abundance. As you watch more of my presentations, the data will overwhelm you. In addition to its cross-feeding benefits, A. muciniphila has direct benefits of its own. A. muciniphila remains in the outer layer of the mucus, so it doesn't adhere to and invade our cells like an opportunistic pathogen could. And the really interesting part is that it possesses a slew of components which can kick off many beneficial cellular responses, which we are just beginning to understand. These components have names like AMUC 1100, 2172, 1434, P9, tripeptide RKH, extracellular vesicles, and diacyl phosphatidylethanol. Upon signaling cellular responses, these components have been shown to increase mucus production and thickness, improving intestinal barrier function, increase antimicrobial peptides, modulate the death of tumor cells, increase the proliferation of intestinal stem cells, increase the differentiation of Paneth goblet cells, activate the differentiation of regulatory T cells, regulate serotonin levels, increase the anti-inflammatory interleukin 10, improve glucose tolerance, improve insulin sensitivity, reduce insulinemia, reduce total cholesterol, and reduce plasma LPS. These are all good things. You're now just beginning to understand the complexity of your microbiome. You are only scratching the surface. Here you can see A. muciniphila at work in an in vitro study. When various butyrate-producing bacteria are cultured with mucin, they fail to grow because they don't possess the enzymatic machinery to access the substrates locked by bonds they can't open. But when these same bacteria are co-cultured, grouped together with A. muciniphila, the sialidases, fucosidases, and other enzymes present in the repertoire of A. muciniphila liberate these fuels, and the abundance of these other bacteria grows significantly, as shown within the blue rectangles, and the butyrate production does the same, shown in the purple rectangles. So now let's see what A. muciniphila can do in humans, not in vitro, while considering what we've covered thus far. Here we have a fascinating study from 2019 out of Belgium. Overweight or obese subjects with metabolic syndrome, but who are not diabetic and had normal hemoglobin A1C, were grouped into three cohorts. Those to receive a placebo, those to receive live Akkermansia muciniphila, and those to receive pasteurized or dead A. muciniphila. After 3 months, believe it or not, the pasteurized A. muciniphila had dramatic impacts, significantly improving insulin sensitivity, reducing insulinemia, reducing total cholesterol, and reductions in plasma LPS, like polysaccharide, which is a microbe marker I discuss frequently. Interestingly, in the live A. muciniphila group, there were no appreciative changes to the overall microbiome. In other words, the introduction of this bacterium did not result in significant shifts of other inhabitants. Now, the dose was 10 billion bacteria per day, and given the many things this bacterium does in the gut, you'd expect other bacteria to either increase or decrease, but they didn't. However, the true lesson here is that of signaling. A dead bacterium is able to plug into, or better said, be read by, your own cells and kick off a set of health-promoting signals that your body carries out. Of course, this is transient only while taking the supplement, which is why you want the live bacterium in your gut, and you want to feed it, which brings me to the commercially available live Akkermansia muciniphila product in the marketplace. It should be noted that their dosing is only 1% of the dosing used in the study. So you could try that probiotic, or you can try to increase your levels by feeding the A. muciniphila, which is probably already present in your intestines, as most people are shown to test positive for its presence. You're not going to ingest mucus to feed it, but you can feed the butyrate producers in the microbiome, who can increase mucin production, which will in turn feed A. muciniphila. Or you can ingest phytochemicals, which, as we've noted, have been shown in a number of studies to increase its abundance. Also worth pointing out is that if components of this dead bacterium can kick off such potent responses in your body, then don't you think that other bacteria, both good and bad, can do the same? And they do. There are whole or parts of bacteria in every part of your body. You don't want a leaky gut releasing large amounts of components from bad bugs into your circulation, chronically ramping up your immune system in every part of you. That's a recipe for disaster. So how does a dead bacterium kick off responses? Well, here we see muciniphila up close. You can see short hair-like structures on their surface. These are called pili. They contain a lot of a protein named muc1100. Various components of the immune system sense various components of bacteria. So muciniphila resides in the outer mucus layer, not adhering to and invading our cells, but via muc1100's interaction with what's called toll-like receptor 4, beneficial cellular functions are initiated, which is all about signaling, another topic I mentioned frequently. One of the consequences of this muc1100 interaction is the increase in the production of what's called interleukin 10. It's a well-recognized anti-inflammatory cytokine, and muciniphila appears to increase its presence on an equal scale as that of the superstar of the gut, Parkinson. Again, inflammation is crucial to survival, but we don't want chronic levels of it. The body needs to know when to pull on the reins. Otherwise, a host of bad things can happen, to include autoimmune diseases, negative outcomes on brain health, cancer, and more. If we look at all the conditions/diseases I've analyzed over the years, we get this chart. These data points come from original research papers examining the human fecal microbiome in healthy controls as compared to those with a given condition. The y-axis represents the number of studies where a significant difference was found between the two groups. Green indicates the abundance of muciniphila was higher in healthy controls, a good thing, while orange indicates that the abundance was higher in the respective disease cohort, a bad thing. As you can see, the color coding is quite variable. In other words, the benefits of muciniphila are conditional. For example, in IBD, inflammatory bowel disease, IBS, irritable bowel syndrome, and obesity, we see that muciniphila is always higher in healthy controls within a fair subset of trials, especially true for IBD. While in colorectal cancer, CRC, and Parkinson's disease, it is always higher in the disease cohort, for a respectable number of studies. We see strong correlations in autoimmune disease for muciniphila to be beneficial, as it is green in all but one study when a significant difference was found. Whereas in COVID, it was curiously higher in COVID subjects in 5 out of 8 studies when a significant difference was found. So how are we to translate this? Let's take a look at CRC colorectal cancer first. This study out of India gives us some classic results with a number of other usual suspects often seen in colorectal cancer. The green rectangles highlight the significant decreases in health promoters in CRC, like Bifidobacterium adolescentis, E. rectale, and F. parasitii, all classic findings. The orange rectangles highlight the main bad actors consistently seen significantly higher in colorectal cancer, to include in this study. The blue arrow points out muciniphila. This is one of the 7 studies from the previous slide I could have shown you. I used 68 data sets for my colorectal cancer meta-analysis in total. So muciniphila is far from being significantly higher all the time. But when it is different, it's always higher in colorectal cancer. But why is a supposedly generally health-promoting bacterium so consistently high in colorectal cancer? We don't know. We can only theorize, but that's a bit problematic, as we'll now see. You can make the argument that since muciniphila is a mucin degrader, and mucin integrity is compromised in colorectal cancer, that it's simply taking advantage of the easy availability of its prime fuel source. And that would be a good theory until we look at the data for ulcerative colitis, which is the classical case of compromised mucin. The results are the opposite. If our theory were true, muciniphila would be feeding away on all the comprised and mature mucin in ulcerative colitis. But instead, when a significant difference was found between UC subjects and healthy controls, in all 12 of those studies, the healthy controls had a significantly higher abundance of muciniphila as compared to the ulcerative colitis subjects. The UC data makes sense, as the mucin coverage in UC is scant and immature, although readily available for degradation. But why is it the opposite in CRC? We don't know. The 8-digit numbers at the bottom are the PMID numbers for all these studies in case you want to verify. Another curious thing about Akkermansia muciniphila is that it seems to be able to survive well in a high pH environment. This is relevant for colorectal cancer, as oftentimes the luminal pH in CRC is quite high. As we see here in Figure 2, the green circles highlighting two amazing health promoters which are negatively impacted by pH. Their counts fall off a cliff with rising pH. This is a well-proven phenomenon in studies. In general, the health promoters favor a lower or more acidic pH. Therefore, in the severely dysbiotic state, such as colorectal cancer, you will generally find their abundances significantly reduced. But a higher than desirable pH is common to many conditions, not just CRC. The truth is that we just don't know at this time. Is A. muciniphila actively causing problems? Is it part of the root cause? Is it guilty by association? We can see within the orange circles the fact that A. muciniphila functions perfectly fine at higher pH. This is something usually attributed to opportunistic pathogens. Therefore, in the spirit of do no harm, I do not recommend the use of this probiotic in colorectal cancer. You can also make the argument that in colorectal cancer, there are dietary changes, such as the reduction in feeding. And data does show that more often than not, reduced food intake tends to increase A. muciniphila abundance. But only a handful of the cancer studies have very ill subjects. And there is also reduced food intake in both Crohn's disease and ulcerative colitis as well. But the picture is the opposite, as we saw earlier. Here is one of the ulcerative colitis studies I used in the previous chart. As we can see here, the colonization rate in Figure A, which means whether it was even detectable at all in the microbiome, was far higher in healthy controls as compared to those with IBD. And when it was detectable in IBD subjects, its abundance, the amount of real estate occupied, was significantly lower than that in controls. Now let's look at the taxa from the 26 Parkinson studies I used for my analysis. These taxa were most consistently shown to be significantly altered in Parkinson's disease, the microbial keystone fingerprint, if you will. In our first column, we see that in 13 studies, half of the studies in my meta-analysis, when a significant difference was found, A. muciniphila was always higher in the Parkinson's subjects as compared to the healthy controls. Why is that? Again, no one seems to know. We do know that Bifidobacterium, Lactobacillus, and Enterococcus all metabolize the popular drug L-DOPA, and so their abundance is consistently significantly elevated in Parkinson's because you're feeding them the substrate they grow on. But there's no data to my knowledge to show that A. muciniphila feeds on any Parkinson's drug. So once again, for those with Parkinson's, I do not recommend the use of A. muciniphila supplementation, just to play it safe. We just don't know what's going on here. Now here's another twist. In my previous slide, when I compiled my meta-analyses for all the major conditions, I did not have this data for healthy aging because it's not truly a condition. When we compile the data from the 9 centenarian studies and the 13 frailty studies I use, we get an excellent idea of the aging microbiome. But again, this includes centenarian data, which is a mix of the aged microbiome, a bad thing, but also a part of the secret sauce to longevity. And that's a good thing. So for example, we can see that the healthier controls had higher levels of classic health promoters, like E. rectale, Phytochemical, and Rosedoria. And the aged and frail had higher levels of Agartella. These are all things to be expected. But to the eye of an expert, some of the others provide clues to longevity. Allostipes and Odoribacter are health promoters, but are significantly higher in centenarians versus young healthy controls. And so is Akkermansia muciniphila. This is not what you'd expect to see with normal age subjects versus healthy controls. So these are likely part of the longevity secret. The metabolic benefits of A. muciniphila are strong, but the dementia data at this time doesn't look good for A. muciniphila. So we take the A. muciniphila data here with a grain of salt. Like anything else, we have to consider the individual. It bothers me when people talk in certain terms, whether it's a type of diet, a supplement, an exercise regimen, or something else. There is no set solution for everyone, 100% of the time. This is true for A. muciniphila. If you suffer from one or more aspects of metabolic syndrome or have IBD or IBS, then the data shows that taking an Akkermansia muciniphila probiotic or trying to increase its presence via specific prebiotics is likely a very good idea. On the other hand, if you have concerns about colorectal cancer or Parkinson's disease, then the data tells us that until we learn more, it would be a good idea to avoid Akkermansia muciniphila. If you found this presentation informative, I have many more for free in my YouTube channel and also in the Microbiome University tab on my website, themicrobiomeexpert.com. There you can select from a wide variety of topics. And if you or a loved one are struggling with a disease/condition, I have condition-specific presentations as well, along with their microbiome protocols found within its respective tab on my website.